Hello, and welcome to Go Beyond A&P. In today's episode, we're going to explore something that everyone has probably heard of, hemoglobin. But we're going to go much deeper than you probably did in your anatomy and physiology course. Most students learn that hemoglobin carries oxygen. While that's certainly true, it's only part of the story. Hemoglobin is actually an incredibly sophisticated protein that is involved into several different forms, each with a specific purpose. Some forms are essential for life before birth, others dominate through adulthood, and others still are associated with important diseases or toxic exposures. By the end of this video, you'll understand why fetal hemoglobin is different from adult hemoglobin, how sickle cell disease changes the structure of hemoglobin, why carbon monoxide poisoning is so dangerous, what methemoglobin is, and why your healthcare provider measures something called HbA1c if you have diabetes. Rather than thinking of hemoglobin as a single molecule, I want you to think of it as an entire family of proteins that all perform the same basic job, but in very different ways. So let's begin by looking at what hemoglobin actually is and why its structure makes it such an incredible oxygen transporter. Then everything else we're going to discuss today starts with understanding how one normal hemoglobin molecule is built. So before we talk about the different types of hemoglobin, we first need to understand what a normal hemoglobin molecule looks like. Hemoglobin is what we call a tetramer, meaning that it is made up of 4 separate protein chains called globins. In adult hemoglobin, there are 2 alpha globin chains and 2 beta globin chains. These 4 chains don't simply sit next to one another. They fold into an intricate 3-dimensional structure that allows them to work together. Inside each globin chain is something called a heme group. Think of the heme group as the business end of the molecule. It's a ringed shaped structure that contains a single iron atom right in its center. That iron atom is incredibly important because it's the actual site where oxygen binds. Notice that the iron shown here is in the ferrous or Fe2+ state. This specific chemical form allows oxygen to bind reversibly. If the iron is oxidized into the ferric or Fe3+ state, oxygen can no longer bind effectively. Later in this presentation, we'll see how that leads to a condition called methemoglobinemia. One of the most fascinating aspects of hemoglobin is that these 4 subunits don't work independently. They constantly communicate with one another. When oxygen binds to one subunit, it actually changes the shape of the entire molecule, making it easier for the remaining subunits to bind oxygen. And that communication between subunits is what makes hemoglobin such an efficient oxygen delivery system. This is one reason why carbon monoxide poisoning is so dangerous. Carbon monoxide doesn't simply occupy one oxygen binding site. It changes the behavior of the entire hemoglobin molecule, making it much more difficult to deliver oxygen to tissues. So what actually happens when oxygen binds to hemoglobin? To answer that, we need to watch the molecule change shape. One of the most remarkable features of hemoglobin is that it isn't rigid. Instead, it behaves almost like a mechanical machine that changes its shape depending on whether oxygen is bound. On the left side of the slide is what we called the T state or tense state. This is the form that predominates after oxygen has been released into the tissues. The globin chains are held tightly together by numerous interactions between the protein subunits. In this configuration, hemoglobin has a relatively low affinity for oxygen. When blood reaches the lungs, oxygen begins binding to one of the iron atoms. That first oxygen molecule is the hardest one to bind. But once it attaches, something remarkable happens. The entire protein undergoes a subtle conformation change, about a 15-degree rotation between the alpha-beta dimers. This shifts hemoglobin into what's known as the R state or relaxed state. In this relaxed configuration, the remaining oxygen binding sites become much more receptive to oxygen. In other words, binding one oxygen molecule makes it easier for the next oxygen molecule to bind. This phenomenon is called cooperative binding, and it's one of the reasons hemoglobin is such an elegant transport protein. The reverse happens when blood reaches the tissues. As one oxygen molecule leaves, the protein begins shifting back towards the tense state, making it easier for the remaining oxygen molecules to be released where they're needed most. And this ability to switch between the two structural states allows hemoglobin to load oxygen efficiently in the lungs and unload it efficiently throughout the body. It's a beautifully engineered molecular system. Many disease-causing hemoglobin variants work because they interfere with this normal structural flexibility. Instead of smoothly transitioning between the tense and relaxed states, abnormal hemoglobins may become unstable, polymerize, or lose their ability to carry oxygen effectively. Now that we've seen how normal hemoglobin functions, we're ready to explore the different types of hemoglobin found throughout life and what happens when that normal structure changes. All right, so now that we've explored how hemoglobin functions at the molecular level, let's establish what normal hemoglobin looks like in the healthy adult. When most people hear the word hemoglobin, they're actually referring to hemoglobin A, or HbA. HbA makes up approximately 97% of the hemoglobin found in healthy adults, making it the primary oxygen transporter throughout most of our lives. Structurally, HbA consists of 2 alpha globin chains and 2 beta globin chains, often written as alpha 2, beta 2. But HbA isn't the only normal hemoglobin present in adults. A much smaller fraction, about 2.5 to 3.5%, is hemoglobin A2, or HbA2. Instead of 2 beta chains, HbA2 contain 2 delta chains, giving it the structure alpha 2 delta 2. Functionally, HbA2 transports oxygen just like HbA, but clinically it becomes very important because abnormal HbA2 levels help diagnose disorders such as beta thalassemia. Finally, you'll notice a third form listed on this slide, hemoglobin F, or fetal hemoglobin. Although HbF is the dominant hemoglobin before birth, healthy adults normally retain less than 1%. That small amount usually has no physiological significance, but we'll see later increasing HbF production has become one of the most effective treatments for sickle cell disease. So the normal adult bloodstream actually contains a mixture of hemoglobins, with HbA serving as the primary workhorse and HbA2 and HbF making up smaller proportions. But this raises an interesting question. If HbA works so well in adults, why would a fetus rely only on an entirely different form of hemoglobin? So to answer that, we need to understand one of the most elegant examples of physiological adaptation in the human body, the transfer of oxygen from mother to fetus. One of the greatest physiological challenges during pregnancy is getting oxygen from the mother's bloodstream into the fetus. Remember, the fetus isn't breathing. Its lungs aren't exchanging oxygen with the atmosphere. Every oxygen molecule must cross the placenta from maternal blood into fetal blood. So how does the fetus successfully compete with the mother's hemoglobin? The answer lies in fetal hemoglobin, or HbF. Notice the oxygen dissociation curves on this slide. The curve for fetal hemoglobin is shifted to the left compared with adult hemoglobin. A left shift means one thing. Fetal hemoglobin has a higher affinity for oxygen. In other words, HbF holds on to oxygen more tightly than adult HbA. As maternal blood flows through the placenta, oxygen naturally moves from maternal HbA onto fetal HbF. I like to think of this as a molecule tug of war. Adult hemoglobin is holding on to oxygen, but fetal hemoglobin has an even stronger grip. That stronger affinity allows the fetus to extract oxygen efficiently, even though the oxygen concentration in the placenta is much lower than it is in the lungs. Without this difference in affinity, oxygen transfer across the placenta would be far less efficient, and fetal development would be compromised. So the fetus doesn't receive oxygen because its blood contains more oxygen. It receives oxygen because its hemoglobin is better at capturing it. But what makes fetal hemoglobin bind oxygen more tightly? The answer isn't simply that it's fetal. The answer lies in its interaction with another molecule called 2,3-BPG. So let's zoom in one level deeper and see why fetal hemoglobin behaves so differently from adult hemoglobin. So this slide explains the molecular secret behind fetal hemoglobin. To understand it, we first need to introduce a molecule called 2,3-biphosphoglycerate, or 2,3-BPG. Now, as a side note, I learned this as 2,3-DPG, 2,3-diphosphoglycerate. 2,3-DPG and 2,3-BPG, essentially they're the same molecule, but nomenclature has changed since I learned it, and 2,3-biphosphoglycerate is more accurate than 2,3-diphosphoglycerate. So we'll call it 2,3-BPG from now on. And there's one place in the slide that I later on that I forgot to change it to from a D to a B, and I'll point it out later on. All right, so although you've probably never heard of it before, 2,3-BPG is one of the most important regulators of oxygen delivery. In adult hemoglobin, 2,3-BPG fits into the center of the hemoglobin tetramer. When it binds, it stabilizes the T state, or that tense state, making hemoglobin less eager to hold on to oxygen. So why is that useful? Because once red blood cells reach the tissues, we want hemoglobin to release that oxygen efficiently. So in adults, 2,3-BPG acts almost like a brake. It prevents hemoglobin from holding oxygen too tightly. Now, compare that with fetal hemoglobin. HbF contains gamma chains instead of beta chains, and those gamma chains alter the structure of the binding pocket where 2,3-BPG normally attaches. As a result, fetal hemoglobin binds very little 2,3-BPG. Without this allosteric brake, HbF naturally remains in a higher affinity state. That's the molecular reason its oxygen dissociation curve shifts to the left. In other words, fetal hemoglobin doesn't bind oxygen more tightly because it's special. It binds oxygen more tightly because it largely ignores one of the body's normal oxygen release regulators. This is one of the most elegant examples of how a tiny structural change can produce a major physiological advantage. And researchers developing new sickle cell therapies often focus on reactivating fetal hemoglobin because HbF naturally resists sickling while still transporting oxygen efficiently. Of course, babies don't keep fetal hemoglobin forever. Shortly after birth, another remarkable transition begins. One of the most dramatic changes in human physiology occurs during the first several months after birth. So take a look at the graph. Before birth, nearly all hemoglobin production is directed towards gamma globulin producing fetal hemoglobin. But shortly after birth, something remarkable happens. The genes responsible for producing gamma globulin begin shutting down, and at the same time, the genes responsible for producing beta globin become increasingly active. This process is known as the hemoglobin switch. Over the next several months, fetal hemoglobin gradually disappears while adult hemoglobin becomes the dominant form. And around 6 months of age, most infants have transitioned to the same HbA dominant pattern seen in healthy adults. So why is this important? Because adult hemoglobin is better suited for life outside the womb. Once we're breathing air, oxygen must be released efficiently into our own tissues rather than extracted from maternal blood. This developmental switch also explains an important clinical observation. Babies born with sickle cell disease usually appear healthy at birth. That's because they still have high levels of fetal hemoglobin. Only after HbF declines and HbS becomes the dominant hemoglobin do the symptoms begin to appear. In fact, one of the major goals of modern sickle cell therapy is to reverse this developmental program, to convince adult bone marrow to start making fetal hemoglobin again. And it's fascinating to think that one of our newest treatments works by turning on genes that were naturally active before we were even born. All right, so far, every form of hemoglobin we've discussed has been a normal adaptation. Some help us obtain oxygen before birth, while others become the primary oxygen carriers throughout adult life. But what happens when the instructions for building hemoglobin change? Sometimes all it takes is a single mutation in one gene to completely alter the behavior of this remarkable protein. All right, so let's see what happens when the blueprint itself begins to change. Up to this point, every form of hemoglobin we've discussed has been completely normal. Now we're going to see how a single change in DNA can transform one of the body's most important proteins. Think about every protein in your body, every enzyme, every receptor, every hormone, every structural protein. Each one is built by following a genetic recipe encoded in our DNA. Most of the time, that recipe is copied with remarkable accuracy, but occasionally a single letter in the DNA sequence changes. Scientists call that a point mutation. You might expect one tiny change to have only a tiny effect. Sometimes that's true, but sometimes one small change can alter an entire protein. Hemoglobin is one of the best examples of this phenomenon. So take a look at the figure on this slide. Normal adult hemoglobin contains glutamic acid as the 6th amino acid on each beta globin chain. In hemoglobin S, that glutamic acid is replaced by valine. In hemoglobin C, that same glutamic acid is replaced by lysine. That's it, one amino acid, one position. Yet those tiny substitutions completely change how the protein behaves. Why? Because amino acids aren't interchangeable. Each one has unique chemical properties. Glutamic acid carries a negative charge and interacts well with water. Valine is hydrophobic. It hates water. It avoids water and sticks to other hydrophobic substances. Lysine, on the other hand, carries a positive charge and changes the electrical interactions within the molecule. Those differences may seem subtle, but proteins depend on their three-dimensional structure. Even one substitution can change how they fold, how they interact with neighboring molecules, and ultimately how they function. One mutation produces the rigid polymers responsible for sickle cell disease. The other produces a much milder disorder characterized by intracellular crystals, and mild hemolysis. The important lesson isn't memorizing glutamic acid, valine, or lysine. The important lesson is understanding that protein structure determines protein function. And sometimes one amino acid changes everything. Modern gene editing therapies for sickle cell disease aren't replacing the entire hemoglobin molecule. They're targeting the genetic instructions that control hemoglobin production, allowing patients to produce more fetal hemoglobin instead of relying almost entirely on HbS. So what actually happens after this tiny mutation occurs? Let's follow the chain reaction from a single amino acid substitution to an entire disease. So this slide tells one of the most remarkable cause and effect stories in medicine. Everything begins with a mutation we just discussed. Replacing glutamic acid with valine creates a small hydrophobic patch on the surface of the hemoglobin molecule. When oxygen levels are high, such as in the lungs, hemoglobin S behaves almost normally. But once red blood cells travel into the tissues where oxygen levels naturally fall, something changes. That exposed valine begins sticking to neighboring hemoglobin molecules. One molecule attaches to another, and then another, and then another. Soon, long, rigid fibers begin forming inside the red blood cells. Notice the sequence shown on this slide. As these fibers grow, they push against the inside of the red blood cell membrane. Instead of remaining soft, flexible, and biconcave, the cell becomes stiff and elongated. Eventually, it takes on its characteristic sickle shape. Now think about where red blood cells travel. Many capillaries are barely wide enough for a normal red blood cell to squeeze through. Healthy red blood cells are incredibly flexible. They bend, twist, fold, and then they spring back into shape. Sickled cells can't do that. Instead, they become trapped inside tiny blood vessels. Blood flow slows or stops completely. Without adequate blood flow, tissues become deprived of oxygen. This lack of oxygen is called ischemia, and it's responsible for severe pain experienced during a vaso-occlusive crisis. But the consequences don't stop there. Repeated episodes of vascular obstruction gradually damage nearly every organ system. The spleen, kidneys, lungs, brain, bones, and even the retina can be affected over time. What's truly remarkable is that every one of those complications can be traced back to a single amino acid substitution. One tiny molecular change becomes a cellular problem. The cellular problem becomes a tissue problem. This tissue problem becomes an organ problem, and the organ problem becomes a lifelong disease. That's why I call this the domino effect of sickle cell disease. One of the most successful medications for sickle cell disease is hydroxyurea. Interestingly, it doesn't correct the mutation. Instead, it stimulates the body to produce more fetal hemoglobin, or HbF. Remember what we learned earlier. HbF doesn't participate in sickle polymer formation. So by increasing HbF levels, physicians can reduce sickling, decrease painful crises, and improve the quality of life for the patient. In other words, one of our best modern therapies works by encouraging the body to temporarily revisit an earlier stage of human development. Not every hemoglobin mutation causes sickling. Some alter oxygen binding instead, teaching us that transporting oxygen is all about balance. So far, we've looked at the hemoglobin variants that are relatively common. Now I'd like to introduce you to something most students and even many healthcare professionals never learn about: high affinity and low affinity hemoglobin variants. To understand these variants, let's first think about what hemoglobin is trying to accomplish. It has two jobs. First, it must bind efficiently in the lungs. Second, it must release oxygen efficiently to the tissues. Those two goals are actually competing with one another. If hemoglobin binds oxygen too weakly, it won't pick up enough oxygen in the lungs. But if it binds oxygen too tightly, it refuses to let go of the oxygen when it reaches the tissues. That's exactly what this graph illustrates. Notice the left shifted curve. This represents high-affinity hemoglobin variants. These abnormal hemoglobins grab oxygen very easily, but they don't want to release it. As a result, tissues receive less oxygen than they actually need. The kidneys interpret this as chronic hypoxia and begin producing more erythropoietin, or EPO. That stimulates the bone marrow to produce more red blood cells. Eventually, patients develop erythrocytosis, meaning an abnormally high red blood cell count. Ironically, these patients don't have a problem carrying oxygen. They have a problem delivering the oxygen at the tissue level. Now, look at the right shifted curve. These are low affinity variants. These hemoglobins release oxygen very easily. Now, that sounds like a good thing, but because they don't hold on to oxygen very well, they also have trouble loading oxygen in the lungs. As a result, arterial oxygen saturation is typically lower than normal. Some patients develop cyanosis even though their lungs are functioning normally. These rare variants remind us that oxygen transport is a balancing act. Hemoglobin has evolved to sit almost perfectly in the middle, not binding oxygen too tightly and not releasing it too easily. Because these patients produce excess red blood cells, physicians may initially suspect a bone marrow disorder such as polycythemia vera before recognizing that the true cause is an inherited high-affinity hemoglobin variant. So far, we've discussed hemoglobins that fold normally. Next, we'll look at what happens when the protein itself becomes unstable. All right, proteins are remarkable molecules. Their function depends entirely on maintaining the correct three-dimensional shape. And if that shape is lost, the protein often loses its function as well. Some inherited hemoglobin variants produce exactly this problem. Instead of folding correctly, the globin chains become unstable. Notice the sequence shown on this table. We begin with a healthy tetramer. Each globin chain fits together precisely, creating a stable oxygen-carrying protein. But if a mutation disrupts one of these interfaces, the protein becomes unstable. It begins to unfold or denature. Once that happens, the damaged globin chains start sticking together inside the red blood cell. These clumps are called Heinz bodies. Although Heinz bodies may seem like a microscopic curiosity, they're actually very damaging. They attach to the inner surface of the red blood cell membrane, making the membrane rigid and fragile. And as these abnormal cells pass through the spleen, splenic macrophages recognize these damaged proteins and begin removing portions of the membrane. Some cells survive temporarily, others are destroyed completely. The end result is hemolytic anemia. Unlike sickle cell disease, where the problem is polymerization, unstable hemoglobins produce disease because the protein simply cannot maintain its normal structure. This is another example of the central principle we've seen throughout this presentation. Small molecular changes often produce enormous physiological consequences. Patients with unstable hemoglobins often present with chronic hemolytic anemia, jaundice, splenomegaly, and elevated reticulocyte counts because the bone marrow is constantly trying to replace prematurely destroyed red blood cells. All right, next we'll examine another hemoglobin abnormality, not one caused by genetics, but by chemistry. All right, throughout this presentation, we've emphasized one critical concept: structure determines function. Now we're going to see how changing the charge of a single iron atom can completely eliminate hemoglobin's affinity to carry oxygen. Earlier we learned that oxygen binds to iron only when that iron is in the ferrous or the Fe2+ state. That's a normal functional form of hemoglobin. However, if the iron loses an electron, it becomes ferric iron or Fe3+. This small chemical change converts normal hemoglobin into methemoglobin. Notice the illustration on the slide. On the left is a functional hemoglobin with the Fe2+. On the right is methemoglobin with the Fe3+. That oxidized iron can no longer bind oxygen, but that's only the beginning of the problem. Remember way back in slide 3 when we discussed the T state and the R state, that tense state and relaxed state? One of the remarkable features of hemoglobin is that its 4 subunits don't behave independently. They communicate with one another. When one subunit changes, the others respond. Methemoglobin takes advantage of that same communication system. The oxidized subunits pushes the remaining normal subunits towards the R state, causing them to hold onto oxygen much more tightly. So not only is one binding site completely lost, but the remaining oxygen molecules are reluctant to leave the hemoglobin molecule. So oxygen isn't just carried less efficiently, it's delivered less efficiently. So imagine a delivery truck. One seat is permanently occupied, and the remaining passengers refuse to get off at their destination. The oxygen is present, but it never reaches the tissues that need it. Fortunately, our red blood cells continuously repair this damage. Special enzymes reduce the Fe3+ back to its functional Fe2+ form. Only when those repair systems become overwhelmed or when inherited mutations permanently stabilize that Fe3+ state does methemoglobinemia become clinically significant. One of the clues to methemoglobinemia is a patient who appears cyanotic despite receiving supplemental oxygen. Another clue is blood that has a characteristic chocolate brown appearance rather than bright red. Methemoglobin prevents oxygen from binding. Our next hemoglobin variant does something even more deceptive. It allows another molecule to take oxygen's place. We've now seen how oxidation of iron can prevent hemoglobin from carrying oxygen. But there's another threat that's even more deceptive. Instead of changing the hemoglobin molecule itself, this one simply beats oxygen in a competition for the same binding site. That competitor is carbon monoxide. Take a look at the balance scale on the slide. On one side is oxygen, on the other side is carbon monoxide. At first glance, it doesn't seem like much of a contest. After all, the air we breathe contains far more oxygen than carbon dioxide. So why is carbon monoxide so dangerous? The answer lies in affinity. Carbon monoxide binds to the iron atom within hemoglobin approximately 200 to 250 times more strongly than oxygen. Once carbon monoxide occupies the binding site, oxygen has almost no chance of replacing it. But that's only the first problem. Remember the R state and the T state that we discussed? When carbon monoxide binds to even one heme group, it stabilizes the entire hemoglobin molecule in the R state. That means the remaining oxygen molecules are held much more tightly than normal. So carbon monoxide poisoning creates a double threat. First, fewer oxygen molecules can bind because carbon monoxide occupies their seats. Second, the oxygen that does bind isn't released efficiently into the tissues. So imagine a delivery truck that's already missing half of its packages and then refuses to unload the ones it still has. The oxygen is present, but it never reaches the cells that it needs. This explains why carbon monoxide poisoning can be so deceptive. Patients may appear relatively normal. They may not be obviously short of breath. And perhaps most surprisingly, a standard pulse oximeter can display an oxygen saturation that appears normal. That's because conventional pulse oximeters cannot reliably distinguish oxyhemoglobin from carboxyhemoglobin. Instead, physicians must use a co-oximeter, which measures the different forms of hemoglobin individually. Treatment focuses on removing carbon monoxide from hemoglobin as quickly as possible by administering 100% oxygen, and in severe cases, patients may require hyperbaric oxygen therapy, where increased atmospheric pressure dramatically accelerates the removal of carbon monoxide. One of the classic signs people associate with carbon monoxide poisoning is cherry red skin. While this is still famous in textbooks, it's actually uncommon in living patients. Most patients look surprisingly normal, making a careful history such as exposure to a faulty furnace, generator, or house fire far more reliable than relying on physical appearance alone. So far, we've looked at hemoglobin variants caused by genetics, structural instability, oxidation, and toxic gases. Our final hemoglobin isn't associated with a disease at all. Instead, it has become one of the most valuable laboratory markers in modern medicine. One of our final type of hemoglobin, like I said, isn't caused by genetics. It's not caused by poisoning. Instead, it's created by something that happens naturally inside the bloodstream. As red blood cells circulate through our body, they are constantly exposed to glucose. Normally, glucose serves as the primary fuel for our cells, but glucose is also chemically reactive. Over time, some glucose molecules attach themselves to hemoglobin through a process called glycation. Now, notice I said glycation, not glycosylation. Glycation is a non-enzymatic chemical reaction. No enzymes are involved. Glucose simply attaches itself to the hemoglobin molecule. Once glucose binds, it stays attached for the rest of the red blood cell's life. Since the average red blood cell lives about 120 days, the amount of glycated hemoglobin reflects the average blood glucose concentration over approximately the previous 2 to 3 months. That's why healthcare providers measure HbA1c instead of relying on a single blood glucose measurement. A blood glucose level tells us what is happening today, while HbA1c tells us what has been happening over the past few months. Think of it this way. Checking a blood glucose is looking at like today's weather. Checking HbA1c is watching kind of like a time-lapse video instead of looking at a single snapshot. This makes HbA1c one of the most valuable laboratory tests for monitoring long-term blood glucose control in patients with diabetes. In healthy adults, HbA1c is generally less than 5.7%. Values between 5.7 and 6.4% suggest prediabetes, and above 6.5-ish% confirmed on repeat testing or with other diagnostic criteria is usually consistent with diabetes. And the higher a patient's HbA1c, or often we just say A1c, the greater risk of long-term complications such as kidney disease, nerve damage, vision loss, heart disease, stroke. One of the important limitations is that HbA1c assumes normal red blood cell lifespan. So conditions such as hemolytic anemia, recent blood loss, blood transfusions, or certain hemoglobin variants can shorten or alter red blood cell survival, making HbA1c values falsely low or difficult to interpret. So after looking at all these different forms of hemoglobin, what are the key concepts you should remember? So let's bring everything together. So first I said that I missed one of that DPG versus BPG. And there it is. So that should be a B. Remember that I said 2,3 DPG is the old terminology, 2,3 BPG is current terminology. All right. So at the beginning of the video, I asked you to stop and think about, or to stop thinking of hemoglobin as a single protein. So hopefully by now you can see why. Hemoglobin is really a family of related proteins. Some forms are completely normal and appear during different stages of life. Others result from inherited genetic mutations. Still, others develop when normal hemoglobin is chemically altered after it's produced. Each form changes how oxygen is transported or how well it's delivered to the tissues. So let's review quickly. HbA is the primary adult hemoglobin. It carries oxygen throughout most of our lives. HbA2, it's the minor adult form that becomes important when diagnosing certain blood disorders. HbF allows the fetus to obtain oxygen from mother's bloodstream because it binds oxygen more tightly than adult hemoglobin. HbS results from a single amino acid substitution that causes hemoglobin molecules to polymerize under low oxygen conditions, producing sickled red blood cells. HbC causes red blood cells to become less flexible, but generally produces milder disease than HbS. Methemoglobin cannot bind oxygen because its iron has been oxidized to Fe3+. Carboxyhemoglobin binds carbon monoxide so tightly that oxygen delivery to tissues becomes severely impaired. And finally, HbA1c provides a valuable record of long-term blood glucose control and has become one of the most important laboratory tests in diabetes management. Although these molecules differ in structure and function, they all teach us the same lesson. Small molecular changes can produce enormous physiological consequences. Sometimes those changes allow life to develop before birth. Sometimes they protect us against infectious disease like malaria. Sometimes they cause devastating illnesses, and sometimes they become powerful diagnostic tools that improve patient care. That's the beauty of physiology. Tiny molecular events often determine what happens throughout the entire body. So before we finish, I'd like to leave you with this one final thought. If there's one thing I hope you take away from today's episode, it's that hemoglobin is much more than an oxygen-carrying protein. It's a remarkable example of how structure determines function. Changing a single amino acid can transform a healthy red blood cell into one that sickles. Changing the oxidation state of one iron atom can prevent oxygen from binding. Allowing glucose to attach to hemoglobin creates one of the most useful laboratory tests in modern medicine. Notice that every hemoglobin variant we've discussed can be understood by asking one simple question. How has the molecule changed? Sometimes it's a change in the amino acid sequence. Sometimes it's an oxidation of iron. Sometimes it's a carbon monoxide binding. Sometimes it's glucose attachment. But every clinical consequence begins with a molecular change. These examples remind us that biology isn't simply a collection of facts to memorize. It's a series of interconnected systems where chemistry, anatomy, physiology, genetics, and medicine all come together. And that's exactly what this series is about. Not just learning anatomy and physiology, but understanding the science behind it. So the next time someone tells you that hemoglobin simply carries oxygen, I hope you smile a little bit, because now you know the story is far more interesting than that. You see how one protein can change during development, adapt to life before birth, become altered by inherited mutations, be modified by chemistry, and even provide a window into chronic diseases like diabetes. That's what makes physiology so fascinating. So thank you for joining me for this episode of Go Beyond A&P. Learn it for the exam and understand it for life. So until next time, stay curious and keep learning.